Hi, I am a PhD student in the Department of Psychiatry, working with Professor Ed Bullmore and Dr. Sarah Morgan. And I'm going to talk to you today about a method that I've been working on over the past year for estimating basically the similarity between brain regions, so similarity networks, just using standard MRI data. So we call it mind networks. So before digging into the weeds, I think it's important to just step back and ask the basic question, what is structural similarity and why is this something that we might be interested in? So fundamentally, structural similarity is a very simple concept. If we have a reconstruction of a brain from, say, a standard T1 weighted MRI image, I'm interested in asking the question, if you have, you've parcellated your brain, you have two regions, how structurally similar are these regions, just using the data that we have available? The obvious follow-on question to that is, why is it interesting to actually compute how similar brain regions are? If we have features for each region individually, why do we need to make a network out of it? What more do we gain from that than just looking at standard structural measures? And sort of the motivation behind this is that this macrostructural measure of similarity that we can get from MRI maps nicely onto lower level patterns of biology that we might be more interested in. So for example, this macrostructural measure of similarity that we can get recapitulates known microstructural properties of the brain. So you have higher similarity between brain regions of the same cytoarchitectural class, so between brain regions with similar microstructure. Also in macaques, brain regions that are more structurally similar to each other have been shown to be more axonally connected using track tracing data. So structurally similar regions are more like literally axonally connected in general. And also, what I find very interesting is that regions that are structurally similar also have similar patterns of gene expression. So using the Allen Brain Atlas, the gene expression profiles of regions that are similar are much higher. This is all work that is sort of background work, and it came from a paper that many of you might be familiar with, so morphometric similarity networks. This came out of Cambridge in 2018, and it is sort of the first method for really validating and understanding structural similarity networks. And their approach was quite simple. Basically what they did is they have a brain region or the cortical reconstruction just from output from FreeSurfer or some standard imaging processing software. And then for each brain region, what they did is they defined a set of 5 to 10 structural features, so that region's mean cortical thickness, the volume of that region, the mean curvature in that region, and so forth. And then for each pair of regions, they calculated the correlation between those vectors as a sort of proxy for the structural similarity between those regions. And that has sort of become the method of choice for structural similarity networks and has been used quite extensively since then. So, and it has been shown to be relevant to copy number variation disorders, schizophrenia, to major depressive disorder, and so forth. So it's quite used. And I was interested in this method, and so when I started working on it, though, I realized that there are kind of some technical limitations to how this method is constructed. Specifically, I realized that when you have this cortical mesh reconstruction, what you have is an extremely high resolution representation of the brain. So you have over 160,000 vertices for each hemisphere. And at each vertex, you have many different structural measurements. And what this method is doing is basically, so for the many thousands of vertices within each parcel, it's just summarizing all of them into summary statistics. And then correlating 5 to 10 numbers is inherently a very noisy estimate of similarity. So what I was interested in doing is coming up with a new method that sort of keeps all of that information and estimates similarity in a much more robust way. So that's where the motivation for this project came forward, and that's sort of where mind networks come into play. So the idea behind the method is that rather than representing a brain region by a vector of 5 to 10 summary statistics, let's include all of the vertex level high resolution information there. So define a particular region by a distribution of vertices, And that distribution is a multidimensional distribution because at each vertex you have many different structural features. So the depth at that vertex, the thickness at that vertex, the curvature at that vertex, and so forth. And then the similarity between those regions can be defined by first taking the statistical divergence between regions and then transforming that divergence into a similarity metric, which I call MIND, which stands for morphometric inverse divergence. And so that is sort of the metric that I've come up with, and which sort of sets the stage for the rest of the analysis steps that I'll present. Essentially, the next few slides are going to be me going through a whole bunch of different validation steps that I have performed to sort of get a sense of what these networks look like, how they compare to MSNs, which are sort of the most commonly used method now, and whether our hypothesis that a more robust estimation of structural similarity can map more closely onto these lower level biological principles that we're interested in, whether that actually holds true. So the first thing that we did is just plot some sample networks. The first two, the matrices up there are just these full connectomes. So for a single subject, structural similarity networks from MIND and an MSN, just from a sample subject, and then the degree of these networks, so just the sum over all the edges in a particular region, is shown on the cortex on the right. At the group level, as we would expect, if you just correlate the edges of the group level MIND and MSN networks, they're correlated with each other, as you'd expect, because they're both estimating similarity. But when you map back to the individual level and you sort of see how similar these networks are between participants, so this is using about 10,000 scans from the ABCD data set, MIND networks are much more consistent across subjects. And also, when you include random noise features into the construction of these networks, MIND is much more resilient to the inclusion of just Gaussian noise, basically. The next thing that we did was compare how the group level networks look when you change the parcellation scheme in the brain. So ideally, the group level networks should be relatively consistent regardless of how you decide to chop up the brain, which parcellation you use. So we looked at three commonly used parcellation schemes: the Deskin-Kiliani, a subdivision of Deskin-Kiliani, and the Glasser 360 region parcellation, and looked at how consistent the networks were across those different parcellations. And what we find is that MIND is substantially more consistent across different cortical parcellations. The next thing that we did was look at how well MIND and MSNs can capture known patterns of structural similarity. So the most obvious pattern of structural similarity that we know is that the brain is symmetric. So there should be extremely high similarity between left and right homologous regions, the same region. And so we found that MIND can capture more strongly weights interhemispheric connections, and it also more strongly weights connections between regions of the same cytoarchitectural class. So it maps more closely onto known patterns of microstructure. This is using the standard von Economo breakdown of cortical microstructure. Having sort of established some of the basic principles of how MIND networks and MSNs relate to each other, we were interested in looking at some of the other signals that were originally published in the MSN paper. So, for example, looking at how these networks relate to data from macaques and how structural similarity using these networks relate to track tracing connectivity. We used a different data set that was used in the original paper, and luckily we sort of recapitulated the original result that MSNs are correlated with track tracing connectivity. But across 5 different track tracing connectomes, we found that MIND is more closely related to track tracing connectivity as well. It's worth noting that the correlation in either case isn't extremely high. You know, the maximum is about 0.4. But just comparing the two methods, at least, we do see a slightly higher correspondence. And that held when we constructed these methods, these networks using different combinations of structural features. So when we left leave one out feature framework or leave two features out framework, and we saw that basically the same pattern regardless of which features we included in these networks. The next validation step that we did was to see how closely mind and MSN related to gene co-expression. And I think that was sort of the part of the original MSN approach that I was very interested in. Like, to me, it made a lot of sense that regions that have similar structural profiles would have similar transcriptomic profiles. And so we were very interested to see how that related to the mind approach. And what we did is we downloaded the Allen Human Brain Atlas dataset, which is a high-resolution gene expression dataset that is sort of the only one in existence that has the spatial resolution needed to do this kind of analysis. So it's from 6 adult brains. And for each region in the Deskin-Kiliani parcellation, we had sort of a vector of gene expression values, and then we calculated the gene co-expression as the similarity between those gene expression profiles between each region. So we created sort of a gene co-expression, gene similarity network for the group, or at least at the group level. Then we correlated the gene co-expression network at both the edge and degree level with the group level mind networks and MSNs. And we found what I was really interested to see is like almost over threefold increase in the correspondence between mind networks and MSNs. So indicating a much stronger relationship between structural similarity as measured by this mind approach compared and gene co-expression as compared to MSNs. So ideally indicating that, it is capturing something more, something about how brain organization, the cortex is organized in terms of its spatial gene expression patterns than MSNs. Spurred on by this result, so basically all of this has been analysis of the group level networks until this point, and so we were kind of interested in understanding, can mind actually tell us anything about inter-individual variations, or does it just describe some general organizational pattern? Rather than looking at specific applications to like disorders or development, we decided to kind of take a step back and ask the question, how much of the variance in mind or MSN phenotypes can be explained by genetic influences? And we thought that would be kind of an interesting baseline to establish before moving forward to apply to different disorders and so forth. And so we just conducted heritability analyses to understand the fraction of these network phenotypes explained by genetic influences. And so luckily in the ABCD data set, we have a huge set of twins, which is great for doing heritability analyses. And there's also genetic data, so you can measure SNP-based heritability as well. And what we found is that, we measured the twin-based heritability for both the degree of these networks, so the heritability of each degree, and also for each individual edge in these networks. And then we also did the same thing for SNP-based heritability. And what we found is that for both of these, mind is more heritable than the corresponding MSN phenotypes, so more closely linked to genetic influence, or more driven by genetic influences. It's noteworthy that, compared to something like surface area, which is known to be extremely heritable, it's not as heritable, but ideally offers some motivation as to why it might be interesting to look at mind when looking at sort of applications to other kinds of disorders, which we know to have some genetic component as well going in the future. And there is more to say about these heritability results. I'm just showing the basic comparison plots here. But yeah, those are the primary results that I have so far. In terms of next steps, where we're going, I'm sort of interested in understanding what we can understand about how this structural similarity connectome changes over development, and how it might, what it might tell us about normal and atypical development. And then also, spurred on by the sort of gene expression result, I'm interested in looking at whether MIND can tell us anything about individual level gene expression. So I have a data set of about 200 people that has both in vivo imaging and postmortem gene expression in the DLPFC, and so I'm trying to sort of understand whether we can infer something about within individual gene expression using their imaging data. And then finally, I'm interested in looking at the genetic signatures of MIND networks using UK Biobank. We have a preprint if you're interested. It's there. And yeah, thank you very much for inviting me. To Ed Bullmore, Sarah Morgan, other collaborators, the brain mapping unit, and then the Accelerate program at the Computer Lab.
Thank you, Isaac. Are there any questions for Isaac? Isaac, please.
Thank you very much. I was just wondering if you'd gone back to ask which particular features are most important in finding good network. You reported single drop and dropping to phosphorylation. I'm not quite sure whether that answers the question. For example, if you take your gene co-expression or your psychiatry correspondence as an external measure of validation, what happens when you drop certain features?
Yeah.
Those are really important, which are not so important.
Yeah, so if you can create MIND from just single features alone, and if you do that, all of the single feature networks are all positively correlated with, say, the gene co-expression network. The one that's most strongly correlated is the sulcal depth network. So that one seems to be the one that's driving the results most strongly. But we also do see that as you add more features, you do get slightly higher correspondence. So there is something to be gained by a multivariate approach as well. In one of the downsides of any kind of structural similarity network is that there's always the question of features, and that's always something that's nagging in the back of my mind. And I don't think it's been fully resolved yet, but it is good to see at least that including more does get you a bit closer to the baseline.
There's no point in being single.
Yeah.
You'll benefit from the covariance. So that's the opposite way around. Having all the features and dropping one at a time, or dropping each one separately. How does that affect performance? Is one of them completely redundant with the other four or five? No effect.
Yeah, I include both volume and surface area, and those are basically the same feature, and so I don't think we need to include. I basically included the same 5 features that were used as like sort of a clear comparison to the MSN approach, but I don't think we need to include, for example, both volume and surface area, or yeah.
Thank you. Is there any other question, final question? Okay.
It's time.
Yeah, quickly.
And so how do they correspond to functional networks? Is MIND better than MSN?
Slightly. I haven't spent that much time looking at functional or DTI networks, which would be a natural next step. I've sort of looked at this casually, and the correspondence between structural and functional networks is, about 0.25 with MIND networks and 0.2 or under 0.2 for MSN, so it's not a terribly strong correspondence. Statistically significantly higher, yes, but I'm not going to put too much weight on that because I think they are measuring very different things.
Okay, well,
Yeah.
thank you.
Thank you.
Very interesting.